Okay, so tell me a little bit about hormones and pregnancy. So we'll start with, what is the definition of hormones and how do they act?
The chemical messengers.
Good.
What was the other question?
How do they act?
How do they act? I think they attach or bind to. Don't recall.
Make sure, guys, when you come into class, you revise your content, okay? Because I expect at least you know the basics. And if you can't answer my questions at the beginning of class, you're straight away sitting at a non-optimal level of understanding to get to a band 6, right? So every lesson when I ask you all questions, they're questions that if you're answering it well, and if you progress to the next class and you continue doing that, by the end of the content, at least from a content point of view, you're at least at a band 6, okay? And it just comes down to fine-tuning from exam questions. So make sure you're revising more consistently. So Matthew, on to you. How do hormones act?
Hormones act by binding to chemical receptors. It first starts in the hypothalamus, where it releases gonadotropin that affects the pituitary glands, that then releases gonads and causes the ovaries and testes to release LH and FSH, progesterone and estrogen.
Okay, good. Yeah, so just make sure that you clarify. So that's an example, right? So generally, when I refer to how hormones act, they act by binding to the nucleus and genes and upregulating protein synthesis. That's one of the main ways they act. They can also act by changing ion channels, so allowing ions to move in and out of cells, which then causes cell signaling pathways. But key point here is that hormones are chemical messengers, travel in the bloodstream, and they bind to receptors, and they change usually the level of protein synthesis and gene expression. Very good. Now, Matthew's also highlighted the hypothalamic pituitary gonadal axis. The only thing I will add is it's GnRH that's released from the hypothalamus, so right in the center of the brain, and that will cause a release of LH and FSH from the pituitary glands. So they're not called gonads. Gonads are a structure. They're called gonadotropins. Tropin meaning changing the gonads. And that's exactly what LH and FSH do. They go and travel in the bloodstream to the ovary. And so can you tell us a little bit about what happens to the ovary? What hormones does the ovary release, Sarah?
Progesterones and estrogens.
Good, right? And Rithani, can you just remind the class, what does estrogen do? What does progesterone do?
Estrogen thickens the endometrium for implantation.
Very good.
Progesterone helps with the uterus not contracting.
Very good, right? Prevents uterine muscle contractions. Very good. If you understand this, you're already at the band 6 level, right? Now it's about getting to strong band 6, especially for your trial exams. And for this, you need to understand this little graph here, right? And let's talk about the main things that you need to understand about this graph and quickly recap that. And I'll recap through questions with you all. So starting with Alyssa. Alyssa, how many days is a normal reproductive cycle? And in which date of this cycle do we get ovulation?
Ovulation is at 14 and the full cycle is 28 days.
Perfect. And your mic is working amazingly. I love that. Great. So 28-day cycle, day 14 we have ovulation. The next thing you need to know, Sarah, is what hormone causes ovulation. If you look at this graph, you'll see one hormone has a prominent spike at day 14. What is that hormone that triggers ovulation?
LH.
Good. Very good. They love testing you on this. LH triggers ovulation, right? That's the next most important thing. Now it's all about a balance of possibilities, right? After ovulation, two things can happen. Ed, what are the two events that could happen in the female reproductive cycle after ovulation?
Fertilization and no fertilization.
Exactly. Perfect, right? So Ed, take us through the most likely path. Let's say there is no fertilization, right? So we get ovulation at day 14, right? And this is triggered by LH. So take it away. What happens if there's no fertilization?
There'll be no implantation either, and
very good.
What happens?
Yeah. And what happens following that?
The endometrium sheds.
Very good, but there is a linking point between that, right? If there's no implantation, why does the endometrium shed? This is very important. You need to know the name of this one hormone that is released by the embryo on implantation. This one hormone sets into the set of events to cause a 9-month pregnancy, birth, and creation of a new organism versus the cessation of that ovulatory cycle and the initiation of a second cycle, right? So maybe I'll go back to Atul. So what is the hormone that's released by the implanting embryo? What is the name of that hormone? Matthew, we'll move on to you. Atul, I'm going to ask you a couple more questions because I need you to answer in a more prompt, timely manner. So Matthew, what is a hormone that's released on implantation of the embryo?
HCG.
Very good, beta HCG, right? So implanted embryo releases beta HCG. If there's no implantation, there's no beta HCG. And so then what happens to the release of estrogen and progesterone by the ovary, Matthew? If there's no beta HCG, does the ovary continue to release estrogen and progesterone?
No.
No, so we see this drop. Perfect, right? And if you know what estrogen and progesterone do, you will know what a drop in estrogen and a drop in progesterone does. So Alyssa, back to you. Estrogen thickens the endometrium. What would a drop in estrogen do, Alyssa?
It would cause the endometrium to basically shed.
Yeah, it'd cause it to thin. And it's mainly the progesterone, everyone. Because what happens is progesterone prevents uterine contractions. So if you have a drop in progesterone, you're going to get massive uterine contractions. And think about all this muscle contracting around this blood vessel. When all this muscle, the muscle is arranged in a spiral manner, when it contracts around the blood vessel, what happens is there is no blood flow to the lining of the uterus. If there's no blood flow to the lining of the uterus, it begins to shed. Any part of the body that does not receive blood, does not receive oxygen, does not receive glucose, it will die. And when it dies, it all sheds. And that is what menses or menstruation is, right? It's the uterine lining removing and shedding all those nutrients and wastes because it knows that pregnancy is not going to occur. It's pretty interesting. The body talks to itself, right? And you're not even aware of this. Yet this is happening every 28 days in the female reproductive cycle until the age of about 53 where menopause occurs, right? It's all very tightly controlled by hormones. Very good. Now let's go through the other possibility very quickly. Edward, what would happen if there was fertilization? So once you know the most likely possibility, the less likely but more interesting possibility is it's very logical. So if there was fertilization, Ed, what happens 7 days after that? Edward?
7 days after fertilization.
Yep.
Implantation.
Good. And then what hormone is released?
Beta HCG.
Good. So what happens to estrogen and progesterone levels, Ed?
They increase.
Good, right? So therefore, estrogen and progesterone remain high. And guess what, guys? This is literally everything you need to know about hormones and pregnancy. I'm going to talk about hormones and birth, but in terms of hormones and pregnancy, just to recap everything for you, if you remember that hypothalamic pituitary gonadal feedback loop, you remember what estrogen and progesterone do, and you remember these two pathways, you'll get full marks every time in every question with hormones and pregnancy. Okay, so break down this huge dot point into very digestible pieces of information. Does anyone have any final questions? Good. Good job. All right, what we'll do now is we will jump down to talking about hormones in birth. So you can do these questions in your own time about flowcharts, but we'll move on. And you can see here is an additional HSC question, which is testing you about beta HCG levels and how they change during the course of pregnancy. Now, I'm going to give you all about 10 seconds to tell me what trend you observe. During graphing the data, look at this table and tell me, how is beta HCG changing? And I'm going to tell you why. Hit the raise hand icon once you see the trend. Atul, I'm going to ask you a question, so be ready to answer regarding this trend. Good. All right. So Atul, in which week of pregnancy does beta HCG peak?
Week 12. Oh, no, week 8 and 12.
Between week 8 and 12, right? So
Yeah.
me logically looking at this graph, what I see, if I was to just very quickly graph it for you all, starts at 0, it goes up to 185, should be on the y-axis. So by week 8, it hits 185. At week, sorry, again, I'm doing it the wrong axis. It's 185 at week 8, and then at week 12, it's at 185, and afterwards, it drops like this. So I'll ask you again, if you had to predict the week number, in which week would you say beta HCG should be at its highest? Week 8, week 12, week 13, what's your answer?
Not sure. Does it have to do with? Oh no, I'm not sure.
Okay. Now,
I'd say-
logically speaking, yeah, exactly, right? It's literally a graph that looks like a parabola. It must have a peak. It does not make sense for it to do this, right? So logically speaking, if it's at 185 at either end, it probably means it's doing this, right? Which means it's probably highest between weeks 8 and 12, most likely week 10. Okay, now I want you all, so now you understand the trend. I want you to think about, why does the graph look like this? And to help you all understand this, Alyssa, can you just remind the rest of the class, what is beta HCG doing at the ovary?
It helps regulate the ovarian hormones during pregnancy, like maintaining the corpus luteum and progesterone so that the endometrium isn't expelled.
Perfect. Did you guys hear the word she said, which was corpus luteum? Now, if I see that as an examiner, I straight away know this student knows what they're talking about, because the corpus luteum is what's remaining of the follicle that contained the ova after ovulation, right? This is a follicle. Inside the follicle will be an ova. Remember, there's hundreds and thousands of follicles. Day 14, that ova is ovulated due to an LH surge, and what remains of that follicle is called the corpus luteum. What does corpus mean? Corporation means body of people. Corpus means body. Luteum refers to hormones. It's telling you that this is a body of hormones or a pool of hormones. The two main hormones being released from here are estrogen and progesterone. And like I told you all, in pregnancy, we want those two hormones to stay high, right? And it's so important to understand this, because to cause termination of pregnancy, you need to give anti-progesterones in the event of, a stillbirth, etc., to get rid of the fetus. So medically speaking, you need to very importantly understand why estrogen and progesterone needs to remain high, right? Those two hormones drive pregnancy. Very, very important for everyone to understand that, okay? But coming back to this point, they need to remain high. But what you're all seeing is beta HCG, which is a hormone that's released from the implanted embryo that keeps that corpus luteum functioning, starts to decline after week 10. Which means after week 10, as beta HCG levels drop, there's going to be a decreasing signal to this corpus luteum. And the corpus luteum, no matter what in pregnancy, is going to degenerate and stop releasing estrogen and progesterone. So can anyone tell me, what structure releases estrogen and progesterone after week 10? Because you all now know from this graph that beta HCG levels drop after week 10, and that corpus luteum, which is responsible for releasing estrogen and progesterone, degenerates. So what structure takes over the function of the corpus luteum?
Is it the placenta?
Very good. A couple of people said it, nailed it, right? And so this graph has actually taught you information that's so important to the syllabus that I think teachers don't often stress, which is after about week 8 to week 10, the placenta takes over the function of that corpus luteum, right? And placenta is so interesting because it is the only time in a human's life, not even a human's life, in a female's life, because males cannot do this, right? Create a whole new organ at a point in life. That is what the placenta is. It's a whole organ. It has so many roles, and we'll talk about that shortly. But the key learning point from this question is that the placenta is responsible for the release of hormones in pregnancy after week 8. It is not the ovary. Okay? Very good. Now, let's talk about the placenta. But before I do that, does anyone have any questions about hormones and pregnancy, what you just learned with beta HCG? What I'm slowly doing is adding a little bit more and more information that is all relevant to the syllabus to help you understand what goes on in pregnancy. Pregnancy is very complicated. Good. Does anyone want to be an obstetrician? So delivering babies, or would consider that as a career path? Hit the raise hand icon if you'd be find it exciting to deliver babies. No one? Okay. I find it scary because a lot can go wrong. And it's also one of the coolest things I ever experienced in medical school, which is being in the operating theater and suddenly seeing new life come into the room. So very rewarding career path. But anyway, coming back to this, let's talk a little bit more about the placenta. Okay. What you're all seeing here, I need you all to visualize this, right? Forget about the whole flow chart that you see around it. Just focus on what you're looking at here. Okay. This is the image that you would expect to see if you need to use graphic imagery so you really understand this. Imagine getting a knife and cutting it straight through my abdomen like this, right straight down, and having a side-on view, right? That is what you would see in a female during pregnancy, right? So I just want to make sure all of us are oriented to this, right? So what you're seeing here, this is the bladder, this is the rectum. That's not important to us. What's important to us are these structures here. Now, can anyone tell me, so this is the vaginal canal. What would this structure be that I've just highlighted there? Edward, what would you call that structure? Very good, the cervix. Good job, Sarah, right? And so what you all see here, this is the uterus and its bulk. And what happens is, originally, I'll just draw a side-on of how it would have looked before. This would have been the uterus, that would have been the vaginal canal, just like this, right? And what happens is the embryo implants here, and it grows and grows and grows in this space enclosed by amniotic fluid. So all around this baby is amniotic fluid. This is the walls of the uterus, right? Now, they have not shown you a very, very important organ, so this is not a very accurate image, but at this point in time, the placenta would be sitting here, ideally, right? Sitting up here, growing out of the wall of the uterus with the baby. Okay, now can anyone tell me, what is this placenta, this whole organ that forms in pregnancy? It's present here. What does it do? What are some of its roles? So, Alyssa, you mentioned the placenta, so I'll let you take it away. What are some of the roles of the placenta?
Basically, it allows things like nutrients and waste to be exchanged between the offspring and the mother, but I think that the blood itself doesn't actually touch.
Very good, right? And it's so important to understand that, because a baby is technically 50% a foreign organism, right? When a person has a child, 50% of the child's genetic information is from a stranger, which is the other parent. And assuming the two parents are not related, that means that child has 50% new different genetic information. So the mother's immune system will attack and destroy the child unless there is a barrier in place between them. And that is the placenta. The placenta serves, number one, as a nutrient exchange, right? So it'll take blood sugar from mum's bloodstream and put it into baby's bloodstream. Okay, so just to give you all a bit of context, you know the belly button that you all have? Feel your belly button. So you realize how real this is. We're not learning some magical, fiction. This is literally you, probably, 2 decades ago, less than that, given your ages. But there will be a vein, the umbilical vein, and an artery, the umbilical artery, coming out of that placenta and injecting into your abdomen. That's what was cut. And what's left of that cut section of the umbilical cord is the belly button, right? So what you need to realize is sugars, nutrients go from mum's bloodstream across the placenta. The placenta is tissue. It's like muscle. That's how you think about it. So it's a pink organ. And so all the glucose will travel from mum's bloodstream through that umbilical artery vein into the baby's bloodstream. And in that process, mum's blood and baby's blood never mix. If they did mix, mum's immune system would destroy the baby's blood and the baby would die. And we actually look out for that during pregnancy. In every single mother's pregnancy, we make sure mum and baby's blood never ever mix. And if they do, it is such a big emergency, we have to deliver the baby. Even if it's 21 weeks, even if it's preterm, it is such a serious medical problem, right? So very important to understand the placenta is a nutrient exchanger. It never allows, it's an interface. It never allows mother and fetal blood to combine, okay? So I'll be writing all this down for you guys. So number one, it's a nutrient exchanger. And the main nutrient I want you all to mention is glucose. It serves as a structural interface, meaning it's physically separating maternal and fetal blood. What else does the placenta do? There's one more thing I would want you all to mention. Think about this. The baby is swimming in amniotic fluid. Amniotic fluid is made from baby pee. So baby pees inside the uterus, and that pee is clear colored urine, and it's amniotic fluid. And then the baby swallows that pee, and weirdly enough, although it sounds so weird, it helps develop the lungs, their gastrointestinal system. And if the baby doesn't swallow, doesn't make urine, doesn't have amniotic fluid, that's a concern. So let me ask you, if you were dunked in a swimming pool right now, would you be able to breathe? The obvious answer is no. So how is this baby getting oxygen? Matthew, any ideas of how the baby is getting oxygen? Because it can't breathe, it's drowning in fluid for 9 months.
Through the umbilical cord, of which diffusion occurs from the placenta into umbilical cord.
Yeah, so literally what happens is, let me draw the placenta and separate it really well for you guys so you can appreciate what's happening, right? The placenta looks like this. It's got 2 separations like this, right? What ends up happening is mum's blood vessels channel in to this side of the placenta. And you see that pink barrier between the 2 sections of blood? And the section of blood here, this is baby's side. Section of blood here is mum's side, and you can see there's a barrier, a tissue barrier separating mum and baby's blood. So what happens is mum's blood goes and deposits. I'm going to write M for maternal blood. It goes and deposits there. And what happens is the oxygen will diffuse across into the fetal blood. And then that oxygen will get delivered via the umbilical veins to the baby. And so the baby never has to breathe for 9 months, which is pretty interesting. The only con is mums can get short of breath a bit in pregnancy due to multiple reasons, but partly also because they need more oxygen. They can also give some to the fetus. It's all very interesting. But the last thing I would say is oxygen delivery. Good. Does anyone have any questions about the placenta? Placenta is an organ, and these are its roles. It has multiple roles. There's also there's way more roles, but for the HSC, you just need to know the main 3. Sugars, oxygen, separates maternal fetal blood. But then what the placenta also does is it releases hormones, right? In the case of hormones in pregnancy, that'd be very important. It releases estrogen, it releases progesterone, it releases so many other hormones. But for year 12, this is all we need to know. Good. Any questions, everyone? Any questions at all? Good, good. So now let's talk about the final thing, right? So you are all masters of pregnancy. You know the exact hormonal changes to cause ovulation, fertilization, pregnancy. You know now what needs to happen with hormones during pregnancy. Remember, pregnancy is about 37 weeks. And a 3.5 kilo fetus, usually 2.5 to 3.5 kilos, is born. My question to you now is, at 37 weeks, what hormonal changes happen to expel the fetus? Because you need birth, right? If birth happens too late, all this amniotic fluid dries up, disappears, and the baby will start to suffocate and get brain damage, and it can die. And if it's born too early, it will not be developed enough to support itself in life. It can also die. The question is, what happens at 37 weeks hormonally to cause delivery? Has anyone learned this at school? Hit the raise hand icon if you've learned about hormones for birth. Good, good. Edward, take it away. What happens to allow birth at 37 weeks?
Oxytocin is released.
Where does oxytocin come from, and what causes it to be released, Ed?
Not sure.
That's all right. Let's try understanding this together, right? So at 37 weeks, you can imagine you have a 3.5 kilo fetus. It is huge. A normal mother gains about 12 kilos during a pregnancy, right? 3.5 is a baby, but there's also amniotic fluid. There's gain in weight. You retain fluids as well. But what ends up happening is, by 37 weeks, this 3.5 kilo big-headed baby is applying force against the cervix. Constant force. And what you need to appreciate is that there are nerve endings at the cervix. It's a sensitive area, right? So when the baby's head, the fetal head, pushes against the cervix, nerves are stimulated. And those nerves send signals at hundreds of kilometers per hour straight to the brain. What were the two hormonal structures in the brain? You guys learned this, right? There's one part of the brain that releases the master hormones, and then a second part in the brain, and then finally you go to the gonads to release estrogen and progesterone. So, Rithani, can you just remind the class, what were the two parts of the brain you need to know for Module 5?
On the hypothalamus and the pituitary gland.
Very good, right? Now, the pituitary is weird. It looks like a sack, and it's got a front and a back to it, right? So I'll do my beautiful artistic drawing of the brain, right? Imagine you chopped your brain right through the center and you're looking at it sideways. That's what I'm drawing for you here. Right in the center, you'll have your hypothalamus. Your hypothalamus will be sitting here. Right underneath it will be this sac-like structure called the pituitary gland. Okay? Now, oxytocin, it comes from the pituitary, and you don't need to know this, but it comes from the back of the pituitary. So you might see in textbooks the word posterior pituitary. Posterior meaning back, anterior meaning front. But all of that is out of our syllabus and scope. For now, I just want you all to understand those nerves at the cervix, which are stretched and excited by the fetal head, they send signals straight to the pituitary, and they cause the back of the pituitary to release oxytocin. Now, what do you all know about oxytocin? I definitely heard of oxytocin before I even started medicine. There's a, you know, they say dopamine is a feel-good hormone. What do you guys know about oxytocin? What kind of hormone is it? So it's, people usually tout it as the love hormone or the affection hormone, right? So when someone feels affectionate towards someone else, caring, oxytocin is released. Now, oxytocin has another function. Hormones don't just do one thing, right? Oxytocin causes contractions of the uterine muscle. So this release of oxytocin is going to cause contraction of the uterine muscle, and eventually the baby will get pushed more. Now, the baby is now getting pushed harder against the cervix. Matthew, what's going to happen to the amount of signals that those cervical nerves, those nerves at the cervix receive if the baby's pushing harder now? Are there going to be more signals, less signals?
More.
More. And if there's more signals, what happens to the amount of oxytocin released from the pituitary gland, Matthew?
It'll increase.
Good. And now more oxytocin is being released, the baby's going to be pushed even harder against the cervix. That's going to stimulate the nerves even more. Even more oxytocin is released. That causes the uterine muscle to contract even more. You guys are seeing the loop, are you not? This is called a positive feedback loop, where everything is constantly stimulating everything else until a final action occurs. And in our case, this feedback loop will keep on going until the fetus or the newborn has been delivered. These are the series of changes in hormones that occur that allow birth to happen. So let me ask you this. Let's say in 7-8 years down the line, you guys have finished medical school, you're working as a doctor, you're on the birth ward, and you've been called because a mum is not delivering a baby on time. There's concerns that the baby will die or get very sick. What hormone will you inject in the mother's bloodstream, or what hormone will you put in the mother's cervix to cause delivery? Edward, what hormone would you pick? Or Sarah? Anyone?
Oxytocin.
Good. That's exactly what we do. If baby's not being delivered, we start what's called a syntocin infusion, which is a synthetic version of oxytocin. And guess what? It works wonders, and it saved so many lives, right? The oxytocin is so important to know, is one of the key hormones that causes delivery. Now, I just want to preface this, everyone. What you just see here, which is a positive feedback loop, is exceptionally rare in the human body and in nature. The body usually, 99.9% of the time, works through negative feedback, in which when a stimulus is exerted, our body will do everything it can to oppose or stop that stimulus. If I'm feeling cold, my muscles will shake until I'm warm enough again, counteracting the stimulus. This is a rare case in time where the stimulus of a baby's head pushing against the cervix stimulates nerves, causes oxytocin release, which pushes the baby's head even harder against the cervix, keeps that pathway going. Very good. Any questions? This has been asked quite commonly in exam questions as an example of positive feedback, because this is the only example you'll ever learn. It's very interesting to compare this with negative feedback, which you'll learn in Module 8. Good. Any questions at all? Okay. Now, you will see some other hormones that are being mentioned during delivery as well. So I'm just going to mention one more hormone, and very minimal about it because this is low yield. But I'm just drawing the placenta again. During birth, the placenta also releases hormones that also help and also cause uterine muscle contractions. And an example of that is prostaglandins. If you'd like to forget this, feel free to, right? It's not going to be an exam question testing you on this, but another hormone that's released by the placenta to cause uterine contractions is this one here. And I have seen it being mentioned in exam sample answers. So if you see it, now you know what prostaglandins are. They're released from the placenta, and they do the exact same thing as oxytocin. Okay? Any questions at all about hormones in pregnancy? Good. You guys have just done what I think is probably the hardest part of Module 5. And they've never ever asked an exam question about hormones in pregnancy. So in 365 days, when you sit your exam and when you get a question on this, just remember that we've covered it quite well and gone through everything. Okay. Any questions at all? Very good. Good job. All right, what I'll do now is we'll see if there's any questions that can be released for you guys to do. See how easy these questions are once you understand pregnancy and hormones. Can you guys hit the raise hand icon once you get an answer to this question? So you can write this down as question 2020 TEC trial paper. They all have different names. This is just one of the trials that students sit. 2020 TEC trial paper. When you get an answer to question 15, hit the raise hand icon. This looks scary to students because some people do the entire HSC never seeing this graph in their life. You guys know you don't need to know everything about this graph. You literally only need to know 2 parts about this graph, and we'll talk about that shortly. Good. Do people have got an answer? Very good. The wording of this question is funky. I know some of you might be thinking, I know a hormone that causes ovulation. Why are they asking about a hormone that's released after ovulation? So really think about this. And the other wording thing is that they ask you, what is a hormone that's realized after ovulation? I think they mean to say released. So think really hard about this. The wording of the question is so important, guys. All right. So, Matthew, what is your answer?
I was gonna say LH because technically it's it happens after, but I'm not gonna go with that answer.
Good. I agree with that, right? So this is the thing. LH causes ovulation. So by logic, it cannot be released after ovulation. Okay? And if the exam answer is LH, you can easily argue this question makes no sense, but it's not LH, right? So now you need to think, well, in this graph, when does ovulation happen? Alyssa's told us. She's told us that the normal duration of a menstrual cycle and the day. So Rathanya, what hormone do you see going up after ovulation? What is the main hormone you see noticeably increasing after ovulation?
Progesterone.
Progesterone. Very good. Right? And that's because progesterone is released after ovulation. And that's because the ovary follicle ovulation happens. And after that, this corpus luteum starts making progesterone. The whole time it was also making estrogen. So that's why estrogen levels are kind of going up, down, changing here and there. But progesterone is only released after day 14 to day 28. So the answer here is progesterone. If the question is what hormone causes ovulation, your answer would be CLH. Okay? Just make sure you're thinking critically as well. Any questions about this graph? Good job. We might move on now. But can you all hit the raise hand icon if you feel quite confident about pregnancy and hormones? You feel like whatever you learned at school, easy to understand, makes sense. Good. Sarah, good. Tanya, good stuff. All right, I think I'm happy to move on. Let's move on to we'll come back to this dot point. This is just about how we used our understanding of reproduction genetics in agriculture. Come back to this dot point. I think for cell replication, I'll need to go through this in much more details. How about we actually go up here and do this dot point here? Okay. Could I get Sarah? Can you actually read out the dot point for us? 1.3.
Evaluate the impact of scientific knowledge on the manipulation of plant and animal reproduction in agriculture.
Good. Now, what does this dot point mean? What do you need to know? What does evaluate mean at all? We've gone through this. What does the verb evaluate mean? Yeah?
Make a judgment.
Okay, so if you just make a judgment, you get 1 mark out of 5 or 6 or 7.
Pros and cons as well.
Very good. And if it's more than 4 marks, what else do you want to do?
Define.
Very good, right? So that's exactly how I break down an evaluate question. So first of all, Alyssa, what is your evaluation? Do you think scientific knowledge has had a big impact on plant and animal agriculture?
Yeah.
Huge, right? It's to the point where right now, bananas never looked the way they do now if you look at the historical evolution of the banana, the plant and tree, bananas are not meant to look like that. They've been genetically modified, reproductively favored and selected over and over again. We've favored and reproduced plants that have increased glucose content, larger fruits. As a result, you end up with this genetically modified yellow banana that we see today. And we see that with all kinds of agricultural produce. Think about milk, right? We've literally found the most optimal, most optimal type of cow or breed of cow that has the most milk production and combined it with cows that have humongous udder sizes. And we've reproduced cows to the point where the cows that we use to make milk today have such large udders and make such large quantities and volume of milk that they get chronic disease and they die early as well, which is not good. So from an ethical point of view, I think it's very surprising and a bit weird that humans very quickly rose to the top of species domination on this planet, and now we use every other species and we modify and change them for our output and benefit. But we're not talking about ethics here, we're talking solely about science and how we use that in agriculture. And I just gave you all a couple of very brief examples, but we'll talk about some that you'll need to mention. So if you ever get this as an exam question, what I want to hear is a judgment that there was a huge impact of scientific knowledge on the development of plant and animal agriculture, right? Now, some examples that I'll tell you, I'm just gonna talk about it. Your full notes are here, right? But I'll draw it and talk about it as well. Okay. So here's a piece of knowledge that fertilization occurs in the fallopian tube. Can anyone tell me what is artificial insemination? What is this technology or behavior? We do it with cows. We inseminate Canadian Jersey cows with spermatocytes from Australian sires. And so that increases beef muscle volume and the amount of beef you can make, but also increases milk production. So can anyone tell me what artificial insemination is? Sarah, do you want to give it a go? What is artificial insemination?
So artificial insemination, it doesn't happen naturally. It's actually, it's done by a medical process. So like IVF.
Yeah, close, but it's not doctors that do this. We don't go to cows and we don't do IVF on cows. By the way, IVF is about $20,000 to $30,000 per cycle of IVF you do. So it's a very expensive process, and that's with humans. This is done by agriculturalists. So what they do is they find a cow. I'm going to save time, so I'm going to get my artistic drawing of a cow. Okay, that's good enough cow for us, right? So you obtain spermatocytes from a cow, you store it, transport it across large geographical barriers, such as from Europe to Australia. You can fly all the spermatocytes frozen, or you can move by train or ship. And then we go to a second cow species. This is the male cow with the spermatocytes. And for the second cow species, we use a pipette. You've all seen a pipette, right? I'm sure you've all used little pasture pipettes where you squirt little bits of water, right? And you inseminate the spermatozoa inside the vaginal canal or close to the cervix of the female cow. And then what happens is spermatozoa swim all the way up and they fertilize in the ova that cow has ovulated in that time point. Okay, so that is artificial insemination. We inseminate female cows using spermatozoa from male cows. Okay, and the scientific knowledge that agriculturalists use for this is, first of all, the location of fertilization, but also hormones in pregnancy. Let me ask you all, what hormones would you give a female cow to force ovulation. And I know some of you might be thinking this sounds very unethical, this process, and you're completely smack bang right. But this is what happens in agriculture. So you just need to know about it for the still point. So what hormones do you inject in a female cow to cause ovulation? Matthew, what hormones would you select?
LH and FSH.
Very good. You need FSH to actually, grow the follicles, and you need LH later on to ovulate the matured follicles. So Matthew would be spot on there. So hormones associated with ovulation. LH, FSH. And that's allowed us to engage in artificial insemination. The explanation of artificial insemination is all up here, right? You can see transfers frozen spermatozoa across geographical barriers into the female cervix. That's artificial insemination. Okay, that's to do with animals. What about with plants? I want you guys to give this a go. I'm going to give you about 40 seconds to tell me, or maybe a minute, just jot this down, the key points. What scientific knowledge do we use to manipulate plant reproduction in agriculture? Talking about examples. Think about pollination, guys. What science do we use in the process of pollinating a plant? I want an exam answer for this one. When you got an answer, can you hit the raise hand icon? I just want you to evaluate the impact of scientific knowledge on the manipulation of plant reproduction in agriculture. We've spoken about animal reproduction. Tell me about plant reproduction. Hit the raise hand icon once you've done that. Just jot about 2 or so lines very quickly, and then I want you to work on how you articulate your answer more than anything. Okay? Just get some water. Okay, so let me get Ruthania. What would your exam answer be? Or evaluate the impact of scientific knowledge on the manipulation of plant reproduction in agriculture. Give it your best go.
I was talking about getting different traits from different plants to and then clone them in order for them for mass production.
So, right. Now, you mentioned the word cloning. Sure, that's good. But I want you to operate in terms of the parameters of what you know. I haven't taught you about cloning properly yet, right? So I wouldn't worry too much about cloning. But you did learn about pollination, right, Ruthania? Because cloning is complicated. I haven't taught you cutting or grafting. I've not taught you guys about stem cell culturing to make plant clones, and that's how you do it. So in terms of what you know, which is you all know pollination quite well. You know the anatomy of pollination, you know the process of pollination. What science do we use for them? That's what I want to know. So let's go to Sarah. What science do we use for pollination?
I'm not too sure. Is it like from the plant, the anther and the stigma? Is it like that, or?
Tell me more, right? What about the anther and stigma? You can't just throw these cool words at me without explaining. So what does the anther do? What does the stigma do? That's the knowledge, right?
So when the male takes pollen from the anther and gives it to the stigma of the female plant, it results in fertilization.
Good. So that's the knowledge. So if I was to word it for this exam question, I would say knowledge regarding the location and location of haploid gametes in plants, which is the anther, and then the ovary of the female plant. Also, location of DNA and how it's stored, because when we're trying to combine and form a new offspring, the understanding of characteristics are expressed from DNA. Right. And then any other knowledge that we use, everyone, you can literally mention anything. You just need to mention one clear point of scientific knowledge and how we use that in pollination, or artificial pollination to be more accurate. Melissa, any other scientific knowledge we use when we're engaging in the process of artificial pollination?
I just had that scientists can cross-pollinate the different species of plants to get the desired characteristics they want, and I used the example of Australian wheat.
Good. Yeah. Okay. So I'd say that's a pretty good answer, right? So as long as you have some clear example. So my example answer would be something along the lines of the location, scientific knowledge regarding the location of haploid gametes as being in the anther bracket pollen and ovary bracket ova has enabled scientists to use forceps to transfer and combine gametes and produce new offspring plants with variable characteristics. This scientific knowledge has also enabled plants to remove the anthers of plants that are receiving pollen to prevent self-pollination, something along those lines, right? So what I just said was we're going from plant 1 to plant 2. If I'm getting pollen and I'm trying to put it down the stigma style in the ovary of plant 2, we actually cut the anthers of plant 2 because we don't want self-pollination. We are aiming for cross-pollination. Does that make sense, everyone? Good. Either way, all my information is written in this table. I would just say know one example of plant scientific knowledge, how we use that to alter their reproduction. One example of animal scientific knowledge, how we use that for animal reproduction. You can see I've also included examples for plants and animals. Wouldn't worry about that too much for your HSC exams. For your trials, they're like asking this kind of stuff. Use examples to support, to see how much you know. Remember, for the HSC, it's not about how much you know, it's about how well you can apply your knowledge and answer new questions. So up into trials, I would remember examples. So in our case, we said the Australian sire is a bull, right? Getting spermatozoa and fertilizing Canadian Jersey cows to increase milk production. And then here, I've spoken about selective breeding for plants. Now, I just want to pause and ask you all, what's the difference between selective breeding and artificial insemination? There is a big difference. Let's go with Atul. Atul, what's the difference between selective breeding and artificial insemination?
In selective breeding, don't they like do the breeding for particular traits, whereas in artificial, I'm not sure what artificial. Yeah.
Close, but both of them are for favorable traits, right? In artificial insemination, why else would we put so much effort in taking spermatozoa from a cow or a bull from one part of the world, transferring it across to another part of the world to inseminate another cow? The goal is we want favorable characteristics, not for survival, for agriculture. The favorable characteristics for agriculture include milk production, increased beef, volume, flavor, all of that, right? Selective breeding and artificial insemination are different in their method. They both have the same goal, which is they're trying to combine characteristic or favorable traits together. That is the goal of both of them. The difference is, in selective breeding, you get organism 1, you get organism 2, and you put them in very close proximity to one another, and you hope for the best. You hope they somehow become really good friends, mate, have offspring, right? In the case of plants, you put them very close together, and you just hope there is a pollinator that will transfer pollen from the male to female plant. Artificial insemination and artificial pollination, which is a plant version of insemination, involves you literally picking out the male sex cell and transferring it, aka inseminating it, into the female reproductive tract. So for plants, that's getting pollen from the anther, putting it down the stigma, style, and ovary, and making sure that we have fertilization. In artificial insemination, it's getting a pipette and injecting it into the cervix of the female cow, and at the same time injecting the cow with hormones to cause ovulation to ensure fertilization happens. Now, I want to see who's thinking, Ed, when would one be favorable versus another? When might artificial insemination be the better thing or the more ideal thing to do?
Like over selective breeding.
Yeah.
Maybe when there's bigger distance between the two. Something.
So let me explain it to you guys, right? Selective breeding, when you do selective breeding, you run a risk of hurting the organisms or potentially killing one, right? If you put a bull and a cow together, the bull does not like the cow. One of them will be injured or potentially killed. So there's a risk of harm in selective breeding. Artificial insemination bypasses that because you don't need the two animals together. You just need to transfer their sex cells. What about artificial insemination, Alyssa? What kind of cons would there be to artificial insemination versus selective breeding?
Maybe that it would be more costly.
Very good, right? I would have to hire a technician, right, to sit there, wait for ovulation, confirm that, and then pipette every single female cow with male spermatozoa. And then there's huge costs in transferring all of the spermatozoa across by ship, etc. Right? So very good. See, this kind of logical thinking is what I personally think is way more important to life and doing well than just memorizing facts, which is what a lot of school is, right? So I want you guys to be critically thinking as much as possible. Good. All right, so that is it for that dot point, and that takes us into inquiry question 2. What I'll get you all to do is take a 5-minute break now. We'll recap at about 3:18, 3:19. But before we do that, does anyone have any questions at all about what we've learned, about their assessment tasks, Okay, good. All right, let's recap at about 3:18, 3:19. Okay, so get up, stretch, walk around. Look around as well, because you've all been just looking at a screen, and get refreshed so we can get back to class. Okay, when you're all back, can you hit the raise hand icon? We'll jump back into things. Okay, good. Has anyone had anything interesting happen in their week? Any interesting things you've learnt? Hopefully every week you're learning something interesting, because your whole life, it's a rare time this is the case, but your whole life is school. And it will not be that case after year 12, because you'll be working, you'll be doing some extracurriculars, having a much bigger social life. So it does get much better after this. But anyone learnt anything interesting this week with biology? No? Okay. Well, I guess interesting thing in my life is I finished my last ever day of hospital for this year. That was very good. So I'm starting my last big break. Then next year I'll be starting work in surgery, so it's quite exciting. Anyone interested in surgery? Can you hit the raise hand icon if you ever thought about becoming a surgeon? Ed, good. Good. You're not. I hate it when students say this, which is that I'm not smart enough to be a surgeon. And I think that's one of the biggest misconceptions about surgery. It's much more about hard work, consistency, and putting in the hours and showing up more than who is the smartest. And even among doctors, the person that becomes a surgeon is not necessarily the smartest out of the applicants. It's the person that has the best patient understanding, the person that's most committed, consistent to the practice of surgery, right? So rocking up and being consistent is a really huge part, especially in most careers. Cool. All right, let's start with inquiry question 2. Can someone read out this inquiry question? Sarah, could I get you to read out just inquiry question 2? And while she's doing that, I want someone to be ready to answer the question for me right afterwards.
How important is it for genetic material to be replicated exactly?
Good. Britannia, what's your answer to that question? How important is it for genetic material to be replicated exactly? What's your answer to this question?
I mean, I guess it's depends on what it is. So mitosis had no genetic variation, so there's no change in genetic material, meaning that is the exact replica. So it allows it to well, it won't adapt to any selection pressures.
But I want you to ask yourself, Britannia, do you adapt to selective pressures in your lifetime? Or does adaptation to selective pressures, does it happen over generations?
Over generations?
Over generations. I'm either born tall, short, fair-skinned, darker-skinned, blue-eyed, blonde hair or not. Whether it's an adaptation or whether it's an unfavorable trait solely depends on where I was born, my environment, right? So having slightly darker skin, this is a favorable trait in Australia where there's high UV radiation that can cause cancer, and so I have a slightly lower risk of melanoma. But if I was born in London, unlucky. I just have darker skin. I won't absorb as much sunlight, won't make as much vitamin D, and I'm more prone to bone diseases. So what I'm trying to get you all to understand is mitosis, you don't want variation, right? If you have variation in mitosis, it's not going to change a characteristic for you. What it might do, though, is cause cancer, right? Adapting to selective pressures happens over generations, everyone, and it's something that a whole population does, okay? But I get where you're coming from, Britannia. So mitosis, you do need genetic material to be replicated exactly. That's all. Can you add on to this question? I want us all to contribute a little piece to this huge inquiry question, which has a pretty big answer. So we've said we need it to be replicated exactly for mitosis. That's all. What else can you tell me? how important is it on your end, do you think, for genetic material to be replicated exactly?
Genetic material has to be replicated exactly so that cellular functions can occur like the same in every single cell, and so that these functions occur properly and like no other functions are like void in the cell.
Okay. Okay, good. Ed, what are the functions of the cell that you need to know for this inquiry question? Edward?
Like replication and yeah.
There's more. What else, Edward? What are the cellular processes?
Cell division.
Cell division, replication. Protein synthesis is one of the main cell functions, right? So for HSC, for this entire inquiry question, you need to know cell functions are mitosis, meiosis, cell division processes. In those processes, we replicate DNA. And then another function that cells do is make proteins. All you need to know. Everything else you learned in Year 11, Module 1. And if you know all these things, you know everything you'll ever need to know about cells the rest of your career or life, right? So coming back, let me give you my answer to this question. Okay, I think it is important for genetic material to be replicated exactly in specific scenarios. Okay, so when we're talking about mitosis, if we have genetic material that's incorrectly replicated, that leads to mutations. And mutations can have two effects. They can either be good or they can be bad. When would a mutation be good? Alyssa, what do I mean by a good mutation?
When it ends up being beneficial to the organism.
Good, right? So it's good when it creates variation and is a favorable trait under natural selection, right? If you have a friend who has blue eyes, somewhere along the lineage they had a brown-eyed ancestor, and that ancestor had a mutation, and they passed on that genetic information, and the child was born with blue eyes. That's where blue eyes came from, right? It was an accidental mutation, and it did not affect survival, so it kept on persisting in small percentages in our population, right? When would a mutation be bad? When it's unfavorable trait. And this is where you have to elaborate this, right? When would it be an unfavorable trait? It's when it impairs with cell functions, as Atul mentioned, but cell functions, that's a vague term. I'm writing FNS as functions. Ed has elaborated to us that the functions of a cell include division, and in division, we're also talking about DNA replication. Matthew, what part of mitosis, or what phase, does DNA replicate?
Interphase.
Very good, right? Good job. So coming back to this, another huge function of a cell, and this is so important because you're going to learn this in a lot of detail very shortly, is protein synthesis. In your head so far, you need to understand cells, they divide, and when they divide, their DNA divides, and when they're at rest, they're making proteins. You can think of a cell as doing those two things. You're set for this inquiry question. Okay, very good. So you can see here that it's important for genetic material to be replicated exactly, but it is also important for there to be some errors occasionally to create variation and mutations that are favorable. Okay? So that's why they asked you this question. They would never ask you a question which has a straight as answer, right? There's no point asking silly straight questions. The inquiry question is worded for you to realize it's not always important for it to be replicated exactly, but majority of the time it is beneficial and most optimal. What about meiosis? For now, I'm just going to write meiosis because that's another cell replication process which involves genetic material being replicated. And we'll talk about what happens when there are errors in meiosis, and you'll touch upon that much more in Module 6 as well. Very good. So I think we've already spoken about mitosis. We have not spoken about meiosis, and we spoke a little bit about DNA. So let's quickly recap this, right? Sarah, can you remind the class, what is the relationship between the words chromosome, DNA, and gene? Alyssa, Sarah, what's the relationship between these words?
I'm not too sure how to explain it, but so basically the gene is what we get from our parents, and so it's basically, I actually don't know how to explain it.
That's all right. That's why you're here. You're learning, right? Good. Nathania, on to you.
I think the chromosome contain the DNA, which have the gene, which code for the gene.
And what's a gene, Nathania? That's all right. I just want to see where we're all at with our understanding, right? I don't care who gets this right or not. I just want to see where we're at with our beginning knowledge, and then I'll explain everything. Alyssa, want to give it a go? What is the relationship between DNA, chromosome, and gene? So the others have alluded to it, but we'll get a bit more detail.
A chromosome is like a linear sequence of a gene, and a gene is like a specific part in the DNA that codes for like a certain characteristic, I think.
I liked your second part of your definition. That was good. Just with your first part, that a chromosome is a sequence of DNA that codes for a gene. That's right. But let me help you understand this, right? The chromosome is the structure of DNA. It is how DNA is structured. Chromosomes are made out of DNA. So you see that purple X-shaped structure? If you zoom into it, you see is that rod is actually just made up of coils. Do you all see that? It's like a thread it's like a snake that's coiled around and around itself to make a very long, linear rod-like structure. And if you zoom in even more to the chromosome, you see that it's made up of balls that are clumped together, and around them is the thread. When you zoom into the thread, you reach DNA's double helix structure. It's crazy. It's like we're going to inceptions on inceptions on inceptions, right? But that is what a chromosome is. Chromosomes are made out of DNA. That's the first thing you need to understand. Chromosomes made out of DNA. But chromosomes are made out of DNA plus these ball-like structures. Ed, what are these ball-like structures that also contribute to the structure of a chromosome but are not DNA?
Amino acids.
You're technically right, but you didn't say what I wanted.
Like polypeptides.
Yeah, you're getting closer. Proteins, histone proteins, right? In fact, a chromosome is only 40% DNA, and 60% of it are these clumps of special proteins called histone proteins. Histone just refers to the type of protein. Okay, you don't need to know anything beyond that. Very good, right? Now, finally, we need to talk about what a gene is. A gene has a very specific definition, and I haven't heard that yet, which is completely fine. Maybe we haven't learned this at school, right? When you see DNA, it's like a four-lettered language, right? There's A's, there's T's, there would be another A here, T, T. This would be a, I've stuffed up the letters because I'm trying to color code them. But if that was an A, that would have been a T. If this was a C, this would be a G. So then we have an A, a T, C, a G, C, G, T, A. You guys are getting the gist, right? Now, a section or a sequence of these letters, usually about 20,000 of these letters or more, will code as the instructions on how to make a chain of amino acids. Ed, what is the name for the bond between amino acids?
Peptide bond.
Good, right? So this is amino acids. The bond between amino acids is called a peptide bond. Okay. What do we call this entire structure here? Can anyone tell me? What do you call a chain of amino acids? It has a very specific name.
Polypeptide.
Polypeptide. Poly meaning more than one, peptide meaning the bonds between amino acids. So if you're a polypeptide, you're a chain of amino acids. Now you might be wondering, what's the relationship between a protein and a polypeptide? Does anyone know? What is the relationship between a protein and polypeptide?
Polypeptides just packaged proteins.
Yes, yes. The only addition I would say is that a protein is made up of one or more polypeptides. Usually, proteins can be 3, 4, 5, 6 different polypeptides coming from 6 different genes that combine together to form a complex three-dimensional shape. So one or more polypeptides equals to a protein. Perfect example, hemoglobin. Hemoglobin is an oxygen-carrying protein that makes your blood red, right? It's made up of 4 polypeptide chains, meaning 4 different genes each make one chain of amino acid, and 4 chains of amino acids combine to form this protein. Okay, so a polypeptide is kind of a simpler unit of a protein. Sometimes proteins can be just one polypeptide. Okay, so very good. Any questions so far about DNA, chromosomes, and genes? Okay, very good. So now we understand DNA and how it's structured. We'll go into a bit more detail on DNA a bit later. We've already spoken a little bit about this cell cycle, but I just want you all to remind me what occurs in all of these stages. So again, I'm going to leave this here because I know some of you may have not touched this in a while, so you might not remember. But I'll give you 30 seconds to read this. And in 30 seconds, I'm going to ask you what happens in every single phase of mitosis. Read and absorb as much as you can. If you have any questions, the next 30 seconds, I want you to ask as many as you'd like. And then I ask you the questions. Okay. Time's up. Let's go through it. This is a cell. This is a cell cycle. Also, we'll start with you. What are the different parts or phases of mitosis?
Interphase, prophase, metaphase, anaphase, and telophase, and then cytokinesis.
Good. Which one's the longest phase, Alyssa?
Interphase.
Good. So interphase takes up literally like this percentage of a cell's life, or well, it's dividing, right? And this remaining stage here is the rest of the PMA. So that's just to have a little perspective on the relative time for each. And as Matthew has said, in interphase we have DNA replication. That's all you need to know about the cell cycle, right? Now let's say I've got a 1, 2, 3, 4 chromosomed cell with a nucleus, right? Now, as you all know, even though it's got 4 chromosomes, as you remember, The chromosome is just DNA that's wrapped around histone proteins. What does DNA make, Sarah? DNA can only produce one type of compound or molecule. What does it make?
I don't remember.
What is a gene? What is the definition of a gene?
It's what's the characteristics that are carried on by the parents, that are given to the offspring by the parents.
Yeah, but remember I told you there's a specific definition of a gene. We can't have fluffy ones because that will confuse us. What did we say a gene was? Britannia, can you help her out? What did we say a gene was just earlier above?
A sequence of bases that form polypeptide chains.
Very good. It's just a bunch of letters in DNA or a sequence of bases that will code for one polypeptide. One gene equals one polypeptide. Everyone who did not know this, I want you to get a red pen and write that right now. One of the most important things regarding cell biology and gene expression. One gene makes one polypeptide. Okay, good. So for us to make polypeptides out of the DNA, there needs to be enzymes that are coming along and reading this DNA and making protein. So for that, what ends up happening is in interphase, the nucleus just looks like a spaghetti soup. Even though there are 4 chromosomes, as you all know, it looks like a soup because all of the thread of DNA has been unwound from those proteins and is just freely floating in the nucleus, right? So remember, when I draw it like this in interphase, I'm drawing it incorrectly. This is not how it looks. Let's move on to prophase. We said interphase, the DNA replicates, so one bowl of spaghetti becomes 2. Okay, all this DNA will double in size. What happens in prophase? Let's go to Ed. What happens in prophase, Ed?
The membrane starts to disappear, and the chromosomes condense into an X shape.
Good. One more thing happens. Britannia, what's the last thing that happens?
The fibers.
Good, right? Because the way I remember this is prophase is a preparation phase for metaphase. Metaphase is when all these chromosomes line up in the middle. And they line up in the middle of the cell. So first of all, for them to line up in the middle of the cell, there should not be a nucleus holding all the chromosomes tightly together. So the nucleus has to go. The next thing that needs to be present are spindle fibers so they can attach to every chromosome. Like this. Okay, very good. So metaphase, I remember they line up in the middle and 2 spindle fibers attached to each chromosome. Now, Atul, what part of the chromosome do those spindle fibers attach to? One chromosome. Very good. So that button here, it's a centromere that's holding the original chromosome and its replicated twin. This is, in interphase, when we said DNA replicates, this is the conjoined twin that was made from that replication. It is exactly identical to this chromosome as well. Very good. Extension question. Alyssa, what organelle makes the spindle fibers? Those little boxes. Yeah,
Not really.
Matthew, any clues? Have you ever heard of the organelle? It sounds like centromere, but it's another name.
No, haven't heard it.
It's all right. Has anyone heard it? Edward, what is it? I'm sure you've heard of it before.
Well, just because what you said, centrioles maybe.
Very good. Centriole, right? So the centrioles are right here. Centriole is a structure that makes the spindle fibers. Very good. Good. What happens in anaphase? let's go back to Atul. What happens in anaphase? Atul, quickly. What happens in anaphase?
Oh, sorry. The sister chromatids are pulled away by the spindle fibers to the opposite sides of the cell.
Good, right? I like to think of this very graphically because thinking graphically and thinking in terms of bright colors, shapes, and sizes is how world memory athletes can even remember 52 cards when you show it to them in sequence. We know for a fact nowadays that memory is not about, what your IQ is. It's literally, do you have techniques to remember facts? The way I remember this in anaphase is if you were a chromosome and your two arms are, the two tops of the chromosomes and your two legs are the bottom parts and your body is a centromere, if your friends pulled you really hard in a room, you'll eventually get pulled to the middle of the room if they pull equally. You go to the middle of the room, and then when they pull too hard, your arms and legs would be ripped off your body, right? And that's literally what I visualize as the spindle fibers doing to these chromosomes, right? Quite graphic, but you'll never forget in anaphase, two sister chromatids rip apart from each other, and that centromere in the middle just disappears because it's made out of DNA, it just breaks down. Good. And then what happens in telophase? Remember, now we need to start looking like a normal cell again, right? And then other thing I haven't shown you is that cell start to look like this. So what happens in telophase, everyone? How do you remember what happens at telophase? Does anyone remember a mnemonic or a way?
Opposite of prophase.
Very good. So what happens in prophase, Atul?
The spindle fibers form and
Good, so they disappear. What's
Yeah.
something else that happens at prophase?
the nuclear membrane forms, so it's going to disappear. I mean, no, it's going to form, sorry.
Yes, very good, right? So nuclear membrane disappears in prophase, it's going to form in telophase, right? And the final thing, so those are the main things. Spindle fibers, nuclear membrane as well, key differences. And then you'll start to form, you have your normal chromosomes, right? They don't have the X shape anymore because they're single rods. So since the X shape of chromosomes, you start to see that in prophase. You don't see that in telophase. You would just see 1, 2, 3, 4, 1, 2, 3, 4. It's 4 pieces got pulled to each side. Good. And then you have cytokinesis, the splitting. Now the reason I want to go through this again was now I'm going to take you guys through meiosis. Now, I've forgotten if I've taught you meiosis, but I'm quite sure I haven't. Can you guys hit the raise hand icon if you've already learned meiosis, either from school or from me? Okay. One of us has learned meiosis before. All right. Very good. So that's why I really wanted to go through this, because meiosis, good. So Tool's also learned about meiosis. Meiosis, I think, is a little bit challenging, but once you understand mitosis well, meiosis is quite easy, right? So now, does anyone have any questions about mitosis and the process? So here's a in-focus textbook image that I really liked about mitosis. So you can have a quick look at it and how everything's changing. But any questions at all so far? Good. What I'll get you to do is a quick exam question, and then I'll take you through meiosis. Oh, what ugly questions. But here's the first one. 2 marks. You'll have a minute to write your answers. This is from question 24 from the 2020 TEC. What is the difference between meiosis and mitosis? Now, the way I know this is a crap question is they haven't put a question verb. Every single HSC exam bio question will have a question verb. Trial papers will kind of skimp this out because they're businesses. They're just trying to pump out papers for schools to buy and to give you. But let's just say that this question written better would have been outline the differences between mitosis and meiosis. So take it away. When you get an answer, hit the raise hand icon. Okay. Good. 2 people have got an answer. Let me go to Matthew. What's your answer to this question?
Meiosis is the formation of 4 genetically varied haploid cells compared to mitosis that forms 2 genetically identical diploid cells. Thus, meiosis forms haploid cells, half the genetic information, that are genetically different, and mitosis forms 2 genetically identical diploid cells, full genetic information.
Good. Good. Yeah. The only thing very good. Right. Genetic, varied. Good. You've given me 3 differences, right? Perfect. The only thing I'd say is I want you all to try this, because this was my challenge as well. When I learned so much in bio, I struggled to write everything I wanted in the least lines possible. That is a whole other technique that we'll all work on. That is a good problem to have, because it means you know a lot and you're doing well. But once you master that as well, and this takes a whole year to do, you have very concise answers. And in that case as well, sometimes in those long responses, 1 mark will be for student provides concise answer with no extra irrelevant, erroneous information, etc. Right? Usually with 2 marks, I think 2 lines or even 1 line would be fine. So even the first line you said, Matthew, was perfect. You could have just stopped there, right? Good. Very good. All right. Let's go to question 1 and 3. This is from question 2019 CSSA, Catholic Schools Association paper. Give it a go, guys. The raise hand icon lets you got an answer. Work for 1 more person. Good. All right, right when I said it. Britannia, take us through question 1. What's your answer and why?
A. Because
Good.
yeast undergoes budding, and budding has no genetic variation, so it's asexual. Good.
All right. Birds, what would birds be? External fertilization or internal fertilization, Ed?
Oh, is that me?
Yeah, birds.
Internal.
Good, internal, but they lay birth to eggs, but still internal fertilization. Good. C, Sarah, what's wrong with C and D?
Bacteria is internal fertilization.
Think for a moment about what you said. Bacterial is internal fertilization. Animals do internal versus external fertilization. Bacteria are one cell in size, right? Remember, bacteria, internal fertilization means fertilization inside the female reproductive tract, which is already an organ made out of tissues. Do bacteria have a uterus or fallopian tube? So, Sarah, what would you say is wrong about C? If you had to recap your answer.
I think the type of reproduction.
Can bacteria engage in sexual reproduction, Sarah?
No.
No. So binary fission is asexual, right? The C is wrong because of that. Finally, Matthew, what about D?
Internal fertilization for ferns, they actually do external because they're plants.
Yeah, but you're completely right in calling it external fertilization, but we call it pollination versus. So when you guys use the word external internal fertilization, we're talking about animals exclusively. Does that make sense, guys? If you ever say internal external fertilization with a plant, you'll get marked down for your exams, even though it technically makes sense because fertilization is happening externally. That term is specific to animals. So for ferns, it's either pollination or vegetative propagation, right? That's why D is not. Right. Good. Because if you think about it, pollination is technically an internal fertilization. The fertilization is happening inside the female reproductive tract, going down the stigma, style, and ovary. And it is contributing to variation. It is sexual. The reason D is wrong is because we don't call it internal fertilization for plants. Good. 3. Atul, what was your answer for 3?
D.
Good. So enzyme activity is all enzymes. Atul, what protein transports oxygen?
Hemoglobin.
Good. And which proteins are responsible for movement?
Is it
Yeah,
actin and myosin?
very good. Actin and myosin. Perfect. So your answer is D. Spot on. I'll give you guys a little bit of a harder question. You're nailing this. There you go. A question related to what you learned today. Hit the raise hand icon once you've got an answer. Good. I was about to say I only had 2 people. Sarah, what's your answer for 12? What do you think about 12? If you're stuck, that's completely fine. Just take us through the ones you can cancel out, because I care about that way more than the right answer right now. Which ones don't look right at all to you if you don't have an answer?
A.
So does A look good to you or not good?
No, it doesn't look right to me.
Good. Why?
Because the it says the secretion happens in the ovary, but then the function it stimulates milk production, so it doesn't sound right to me.
Okay, good. So the function doesn't sound right to you. What about the site of secretion? Does oxytocin come from the ovary?
No, the placenta.
Close. Prostaglandin comes from the placenta. Remember, oxytocin comes from the back of the pituitary gland, and it happens because the baby is pushing its head against the cervix. It stretches nerves at the cervix, and those nerves go to the back of the pituitary, and they cause a release of oxytocin. So the pituitary gland would be the right answer for oxytocin. Okay, good. What about C and D? Is HCG released from the ovary, Edward? No, it's not. It's released from the implanted embryo. So if anything, the embryo would be the answer for site of secretion, right? So C is not right because HCG doesn't stretch the cervix. That's what oxytocin does because it causes muscle contractions. So your best answer is B. Very good. All right. Question 14 and 15. Last two questions, and I'll take us through meiosis. Good. While you guys are doing this question, honest raise of hands, because this was always a problem for me. Does anyone find it hard to stay organized and on top of everything with year 12? Maybe you don't feel it yet because it's just the first term. But have you guys found that it's a little bit hard to stay organized for year 12? Yeah, good, because I found it hard. So I think it's very common, if not anything. So what do you guys do to stay a bit more organized with school and your assignments and your homework and everything? You'll feel it much more when exam time comes, because then you have to balance studying for 5 to 6 different subjects. You have to know when do I have to revise maths again versus 2 unit versus bio versus chem, and also allocate more time to your weaker subjects. Because remember, HSC ATAR is an all-rounder game. If you smash maths but you suck at English, you'll get sucker punched in terms of the ATAR, right? So what do we use as tools to stay organized? Matthew, what are your ways to stay organized and on top of things?
I don't really. Besides, I was gonna do a timetable for study, but I didn't end up doing that.
Timetables are hard because things change, right? Sometimes you don't feel motivated, you don't get that work done, and then it changes every other day of that week. So, understandable. Atul, what about you?
Yeah, I don't really do anything either. I just do what I want on.
That's fine. Yeah, look, first time you're 12, it's not going to really matter whether you do it or not. But you start now and it will really help you come trials time when you realize there's like 50, 60 trial papers for every subject. And the person that does more does better. Ed, what about you?
Gonna try to write down everything that I need to do, and then just do stuff that I have to do from teachers and stuff first, and then stuff. But I don't always do that.
I think, Ed, you messaged me a while back about your timetable and I actually really liked it. So, and it's very similar to what I do. So what I do is I use Notion. And on Notion, it's literally limitless in terms of the way you want to create, calibrate timetables, habit trackers, etc. I had a habit tracker and it was really cool, but I stopped using it. But my timetable basically has a weekly timetable on Notion. And it's like a checkbox kind of thing. And every day I put 3 things that I want to get done. Only 3. The 3 most important things. Then all the other boxes are just optional tasks that I need to get done for that day. I need to check it off. And it auto recreates itself every week. So if you guys would like, I'll send you my template for that on Notion. But I'd really, really advise you guys to stick to habits now. The reason being, even more important than all this content stuff you're all learning now, the key thing that's going to determine your success in life, more than this, whether you understand prokaryotic, eukaryotic, is first of all how critically you think, and second of all, the habits that you have in your life now are the habits you're going to have when you're 23, 24, my age as a young adult, and then later on in terms of work. So start finding systems or ways or routines, such as my weekly 3-item, goals plus, weekly agenda every single week, to kind of keep yourself generally intentional with the way you're progressing with life. Otherwise, you just be scattering around whatever's due next. And I see a lot of people doing that even nowadays, including myself. One book I recommend all of you read, I'd say it should be in the school syllabus or curriculum, is the book Atomic Habits. I think that's much bigger and more important homework for your holidays. That is the only self-help book that I found actually does something for your life versus fluffy nonsense. So have a read of that and let me know how you find it, because it's one of the most powerful habit tracking and behavioral change books. Cool. Anyway, coming back to this, question 14 and 15. What's your answer, Matthew, for 14?
My answer is B.
Good. I'd agree with you, right? When you break the single strands, you break the hydrogen bonds, which are very weak. Does anyone remember what temperature you need to break these hydrogen bonds? And I'm not asking it just out of fancy facts. This is directly used in a technology you will be learning in about 2 weeks, 3 weeks time for this module. So when I first taught you about these hydrogen bonds, I told you there's a temperature they break at. Does anyone know what that temperature is? About 95 degrees. At 95 degrees, those bonds break. And we use that when we're amplifying DNA in test tubes in a technology by the name of DNA profiling and PCR, or polymerase chain reaction. You're going to learn about that very shortly, so keep that in the back of your mind for now. Question 15. Britannia, what's your answer?
B.
Good. Can prokaryotic cells have more than one chromosome, Britannia?
Yeah.
No, prokaryotic cells only ever have one circular chromosome. This will always be the case when X is prokaryotic. Never forget that. And the next question I'm going to ask you is, what is an additional piece of DNA in the cytoplasm of a prokaryotic cell? What is the name of another piece of DNA that sits within a prokaryotic cell? So we've got the chromosomal DNA.
Is it RNA?
Not quite. A very good guess. What do we call those little circular loops of DNA that also sit in bacteria and prokaryotes? This is going to be very important for Module 6. Has anyone heard of plasmid DNA before? Can you hit the raise hand icon if you have? Okay. This stuff is really, really cool because plasmid DNA are little loops of DNA that can carry genes that bacteria can exchange between each other, right? So let's say you start taking an antibiotic and this bacteria is about to die. This bacteria can pass on a little plasmid that contains the gene to become resistant and block this bacteria from being killed by antibiotics. Right? So that is how bacteria can evolve so rapidly. They can literally pass genes between each other. We can't do that. I can't give you my genes. You can't give me your genes. Bacteria can do that through plasmids. So remember that bit of information, because we have a whole technology that has a potential to change the world and is doing so right now based on this information. Cool. Let's move on back to the time today. So what we'll do is, what I'll get you all to do for next week is I want you to have a read of meiosis all the way up to page 105. Okay. I'll be asking you questions from the checkpoint sections of 105 for next week. Okay. Make sure that is done. So you're going to have to read through meiosis and also polypeptide synthesis, and in class I'll teach that at a slightly faster pace. Okay? Good. All right, good job, guys. Have a good week. I'll see you next week, and message me if you have any questions or assignment help you'd like, etc. Okay? And for those of you coming to chemistry, I'll see you in a minute.
So could I just speak to you for a minute after
Sure.
class? Thank you.
All right, let me just stop the recording. How can I help, Sarah?
So I haven't received the booklets yet.
Yeah, a couple of students have actually mentioned that. I think what's happened is it's been